Good morning, good afternoon, and good evening, everyone. And welcome to the Scientist.com and Proteogenics webinar titled Overcome the Multiple Challenges of Neutralizing Antibodies in Cancer Therapy Using Innovative Phage Display Technologies. I'm Liam Sanyo from the events team here at Scientist.com, and I'll be the host today. We're very fortunate to be joined by Dr. Conor McCartin, who's an account manager in monoclonal antibody development at Proteogenics. Today, Dr. McCartin will discuss how phage display can be used for human antibody development. And with that, I'm very pleased to welcome Dr. Conor McCartin. Conor, thanks so much for joining us today, and the floor is yours whenever you're ready.
Okay, so thank you, Liam, for the introduction, and thank you to everyone who has signed up for this presentation today. As he said, I'm Conor, an account manager specialized in monoclonal antibody development here at Proteogenics. We are a French contract research organization which has been operating for 20 years now, with a great deal of experience in monoclonal antibody development, and I hope to share some of that experience with you today in my webinar focused on overcoming the multiple challenges of neutralizing antibodies in cancer therapy using phage display technologies. So, first of all, since antibodies are, of course, a very common and useful tool, I will start with a quick poll. While you answer that, I can give you some figures on the growing importance of monoclonal antibodies in the global therapeutics market. As you can see, in the last few decades, there has been a massive increase in the proportion of approved biopharmaceuticals, which are monoclonal antibodies, with over 50% of approved biopharmaceuticals in 2022 being monoclonal antibodies, with over 150 FDA total approved therapeutic monoclonal antibodies as of 2022. The total monoclonal antibody market is set to reach over 200 billion USD in 2023, with the prediction for 2030 being over 450 billion USD, representing a continual year-on-year increase of around 13%. If we look at the market share by type of disease, we see a very clear dominance of cancer and autoimmune diseases, with the rest altogether representing only around 10% of the total market. So, of course, monoclonal antibodies aren't just one type of molecule. We can see the structure of a full-length IgG here, with two heavy chains, each with 3 constant domains and 1 variable domain, and 2 light chains, with 1 constant and 1 variable domain each. This being the most classical form of antibody used throughout probably all applications of monoclonal antibodies. But, especially in development, we can also work with fragments of these antibodies, taking only the variable antigen recognizing part of the antibody, an scFv, or a somewhat larger portion known as a fragment antigen binding part, or Fab. These have the major advantage of being possible to express in functional form in prokaryotic systems, which is useful in many cases in the developmental stage of therapeutics or for the development of diagnostics. Very important also for therapeutic antibodies is the degree of humanness of the antibody. So the first monoclonal antibodies developed in the 70s were via the hybridoma technique and thus were fully mouse. However, this of course caused immunogenicity problems, as injecting mouse antibodies into a human may of course elicit an immune response. The first answer to address this was simply to take the variable antigen-specific segment developed in a mouse and then artificially grafting it onto a human backbone, giving a functional antibody that is only 25% mouse. However, although a major improvement, this can still cause immunogenicity problems. And so a further improvement on this were humanized antibodies, whereby individual amino acids are intelligently changed to make the antibody sequence more human and less immunogenic. But even more recently, we have since developed the capacity to directly generate fully human antibodies for which immunogenicity is just not an issue. Of course, without the need to inject and immunize live humans, which would be a tad unethical. We can see these developments in the types of monoclonal antibodies that are being approved over time. We started out slowly with mainly some rodent antibodies, some chimeric and then humanized antibodies then came online, with fully human antibodies now taking more and more space in the rapidly increasing amount of monoclonal antibody approvals. So the question thus remains, how do we develop these fully human monoclonal antibodies if we are not directly immunizing humans to isolate them? The answer to this is, of course, Phage display. So basically, Phage display is a wonderful technique which won the Nobel Prize for Chemistry in 2018, whereby we can take a bacteriophage, a virus which infects bacteria, and so which is of no threat to humans, and modify the wild-type P3 protein of the phage to form a fusion protein with an antigen-recognizing antibody fragment. Since there is no species limitation, we can modify these phages with entirely human antibody fragments, which we can then use for the development of fully human monoclonal antibodies. In order to construct a phage display library, we can isolate B cells from human donors, extract the total RNA to then synthesize cDNA, then PCR out the variable chain sequences responsible for antigen recognition, and then clone this into a phagemid vector encoding the viral bacteriophage proteins in order to create a library of bacteriophages displaying antibody fragments corresponding to the immune repertoire of the isolated human B cells, which can then be transformed into a special strain of E. coli for use, with quality control assessment then carried out via colony PCR to assess the sequences and the rates of insertion, the in-frame sequences, the diversity, lengths of the CDO regions, and the gene families. One example of such a library is Proteogenics' high-diversity naïve human library, which has been constructed from 368 healthy donors from 5 different ethnic backgrounds in order to maximize the diversity of the library, as there can be significant variability in the immune repertoire of individual humans. This has allowed us to obtain an exceptional library with over 5 times 10 to the 10 unique antibody fragments, with an insertion accuracy of over 97% of the sequences into the phagemids, thus providing a great source of therapeutic antibodies for which no humanization is necessary. So the question then remains, how do we then use this library to get out fully human antibodies? This process is known as panning, whereby we first coat our target, which may be a protein, a peptide, or even a small molecule, to which we then add our phage library representing the human immune repertoire of antibodies, with those which carry antibodies specific for our antigen then binding to the antigen, while the majority of the phages which are non-specific would be washed away, allowing us to identify binders of interest amongst the billions of different antibodies present in our phage display library. The bound-enriched phages against the target can then be eluted at acidic pH and then used to reinfect E. coli for amplification and restarting of the panning cycle. This is repeated for several rounds to continually enrich the phage library against the target to get high-affinity, high-quality binders. The enrichment at the end of each round is followed using an ELISA of the phages against the coated antigen. This is in order to identify the ideal middle ground between enrichment and sequence diversity in order to be able to propose several binders against the target. If we do not do enough rounds of panning, enrichment will not be detectable, while if we do too many, a very select few sequences will dominate the enriched phage pool, thus lowering the diversity of binders obtained. Following the conclusion of the panning rounds, a limiting dilution of the enriched phage pool from the selected optimal round or rounds is carried out to identify monoclonal phage binders corresponding to one sequence, which are then screened against the antigen and sequenced to get the final specific antibody sequences. thus making this a fantastic method of quickly getting a selection of fully human antibody sequences against a target of interest. However, there is one potential problem. If we are talking about getting human antibodies against human therapeutic targets, then the human B-cell repertoire which we use to construct the human Phage Display library will have a limited number of antibodies against our desired human targets. This is due to the natural mechanism of B-cell central tolerance, whereby autoreactive B cells are either edited or killed by our immune system. One way of getting past this in order to have a Phage Display library with a greater quantity of human anti-human antibodies or autoantibodies is by exploiting a breach of self-tolerance for the creation of our Phage Display library. We can do this by taking a pool of B cells enriched for autoantibodies for our RNA isolation, PCR, and cloning. In order to take advantage of this, here at Proteogenics, we have thus created our new autoimmune-based Phage Display library, whereby we have taken PBMCs isolated from 42 patients with a range of autoimmune disorders, and thus an enrichment of autoreactive anti-human antibodies. This library has a massive diversity of 1 × 10⁻11 different clones with an excellent in-frame insertion rate and minimal construction bias due to separate processing of the kappa and lambda light chains, giving a fantastic pool of enriched higher affinity binders against potential human therapeutic targets. However, autoimmune disorders are not the only type of disease which presents an enrichment of human autoantibodies. As cancer represents a shift away from recognized self, the immune system in this case starts to generate antibodies against tumor-associated and tumor-specific antigens in order to differentiate the tumor from the rest of the host. Thus, bearing in mind that cancer is a major group of diseases which represents a large portion of human therapeutic research, these tumor antigens against which antibodies would be enriched in cancer patients represent a highly interesting set of targets for therapeutic research. In order to address this important need, we have thus developed our human cancer Phage Display library, which we have constructed from the PBMCs of 48 patients at different clinical stages of different types of cancer, including breast, prostate, colorectal, renal, lung, melanoma, and acute myeloid leukemia. This library reaches a great diversity of 3.81 × 10⁻10 different clones and represents a very important and very useful pool of binders against human cancer targets. And while these are some of the ready-made Phage Display libraries which we can offer for our pannings, if you require an enriched antibody repertoire against a specific pathology, which we do not have, then the construction of your own personalized Phage Display library with Proteogenics is also possible. We have done this on many occasions, such as for COVID-19, which I will discuss a bit more later. Otherwise, although it is extremely important to have the right antibody repertoire within which to search for your therapeutic antibody, getting out the right antibody from your library is a separate task, which requires its own special know-how and protocols. In the case of anti-cancer therapeutics, in many cases, a specific blocking or neutralizing activity is desired for a functional therapeutic effect against the tumor, either to block the receptor or to block a ligand binding to a receptor. Many such approved therapeutic monoclonal antibodies are growth factor inhibitors, such as this, which can have a direct effect at inducing apoptosis by blocking such interactions. Some examples being trastuzumab, the HER2 binder, cetuximab, the EGFR binder, or bevacizumab, which binds VEGF to impede angiogenesis. Another class of useful neutralizing antibodies are immune checkpoint inhibitors. These are designed to block receptor-ligand interactions between cancer cells and immune T cells, which are used by cancer cells to deactivate the T cells and preventing them from killing the cancer cell in question, with this coincidentally being a discovery which also won the Nobel Prize in 2018 in parallel to Phage Display. Examples of antibodies with such mechanisms are pembrolizumab, which is targeted against PD-1, atezolizumab, which targets PD-L1 on the other side of the same interaction, and ipilimumab, which targets CTLA-4. For these important cases, we have optimized a special protocol to select for these ligand receptor interaction neutralizing antibodies. For this, we carry out a depletion of our Phage display libraries with every round against the ligand receptor complex. With the ligand blocking the site of interest on the receptor, only the phages which do not interest us will bind, allowing us to recover those which do interest us to use as an input into our panning rounds, which we will then repeat for several rounds as explained before. And then at the end, we will also carry out a competitive elution in the final rounds. This means that instead of eluting all of the phages using acid as normal, we remove the phages bound to the target by adding a saturating concentration of the ligand against the site of interest, specifically removing the phages bound to this site and thus enriching against the epitope which we require for our therapeutic effect. This allows us to screen a specifically selected pool of phages whose binding to the site of interest is validated using a competitive ELISA, allowing us to identify binders positive against the receptor but negative against the ligand receptor complex, getting us our therapeutically active neutralizing antibody. And of course, since there are a range of developability parameters which are important for therapeutic antibodies, which may not be evident or measurable at this Phage display selection phase, such as a propensity to aggregate at higher concentrations, we may carry out bioinformatic analysis of the sequences obtained at this point in order to identify which may be the best ones with which to go forward, saving a potentially very large amount of time and resources which could be spent pursuing the wrong candidate. However, yet another property of such antibodies is to be considered which can't be so easily measured in silico. As the therapeutic development process is very long and naturally goes through different preclinical stages with different species models, often passing through mouse and monkey models before entering clinical trials. It can thus be very important for a therapeutic candidate to be cross-reactive against all species which will be used during the development process so that it may be assessed on a level playing ground throughout. For such cases, we can propose a specific co- or cross-panning strategy to specifically select for binders against epitopes which are common to each of the orthologs, with the cross-reactivity of the binding, of course, being validated via ELISA using the phages, allowing us to validate that we have an antibody which can be used at all stages of the preclinical and clinical trial process. However, although all of this may sound a little bit easy, therapeutic targets are often multipass membrane proteins which cannot be expressed freely in soluble form, and so are rather difficult to study and to pan against. Thus, to address this, we also have a solution for the correct native expression of difficult to express receptors themselves. For this, we propose a range of solutions to identify binders against these targets, such as their expression in virus-like particles, micelles, and nanodiscs, or otherwise synthesis and panning using short peptides corresponding to unstructured loops on the proteins. Otherwise, to identify binders against the truly native conformation of the proteins, we may carry out panning and/or Phage screening on the cells overexpressing the target protein itself, ensuring that the binders we get out of the library are relevant for the intended therapeutic applications. So, I will now move on to a case study which will show these techniques that I have described in action. While we can't in most cases share the details of our projects due to the confidentiality we have with our clients, I would like to share you the details of one exceptional cross-reactive neutralizing antibody project where we can, as it has already been publicized. So, during the pandemic, we teamed up with Asim Healthcare and Trident Biopharma Solutions in order to develop a neutralizing antibody cocktail against a range of the COVID-19 strains circulating at that time. It was identified very early on in the pandemic that the receptor binding domain, or the spike protein of COVID-19, bound to a protein expressed on the surface of certain human cells known as ACE2. Thus meaning that antibodies which neutralize the interaction between these two proteins can be used as an effective preventative and curative treatment for acute COVID-19. Step 1 of the project started with the construction of a custom human scFv Phage Display library for our client using PBMCs sourced from a COVID-19 survivor, giving an excellent diversity, insertion accuracy, and sequence accuracy. We then took the recombinant receptor binding domain of the SARS-CoV-2 alpha variant and recombinantly produced human ACE2 in order to carry out our Phage library depletions. With cross-panning against different variants, followed by a competitive elution with ACE2 and a competitive ELISA screening to identify the competitive binders which were of interest to us. Once a panel of 8 binders was identified, we sequenced the clones and expressed them as full-length recombinant human IgGs by using our high-yield proprietary mammalian XTEN-CHO system. We then assessed the activity of this panel of binders using our own surrogate virus neutralization test, which we developed ourselves at the start of the pandemic in order to identify neutralizing COVID-19 binders. This test involves a HRP conjugated human ACE2, which will bind the COVID-19 receptor binding domain. If antibodies binding the COVID-19 receptor binding domain are present, then this will block the access of HRP conjugated ACE2, resulting in a lowered ELISA signal caused by the neutralizing antibodies. Several clones, such as the example shown here, gave highly promising cross-variant inhibition results using this SVNT test, with the values here being the percentage inhibition against the recombinant receptor binding domain of each variant circulating at that time. This allowed us to confidently move forward with tests against live COVID-19 virus. A plaque reduction test, plaque reduction neutralization, or PRNT50 test, was thus carried out, whereby live virus is mixed with the antibody, then placed on plated cells. A reduction in the number of viral plaques, representing a reduction in the infectivity of the virus, and thus a successful neutralizing effect, which may be therapeutically relevant. The result showed a fantastic neutralization effect of the antibodies against all of the variants tested, with the inhibition of viral plaque formation here being plotted against the concentration of the antibody used, showing the great efficiency of our depletion, competitive elution, and cross-panning strategies to obtain neutralizing binders which are relevant in a therapeutic setting. Thus, to summarize and extrapolate these principles we have discussed to their potential for the identification of therapeutics for cancer and other pathologies, we at Proteogenics have our cancer and autoimmune libraries, which are a strong basis for the identification of therapeutic antibodies, as they capture an important enrichment of human autoantibodies. As shown for COVID-19, we can also supplement these libraries with specially constructed libraries again with strategic donors from desired target pathologies. We have a highly effective protocol for the identification of clinically pertinent blocking or neutralizing binders, with depletions to remove binders against non-interesting epitopes, competitive elutions with saturating concentrations of a binder against the site of interest, and cross-panning to select for desired cross-reactivity. And for difficult to express membrane-bound targets, we have a range of panning solutions so that we may pan against them in their native form and thus clinically relevant conformation, which is essential if we are looking for a therapeutic targeting antibody. However, although I have talked almost exclusively about human therapeutic antibodies, these are not the only option for human therapeutics. Camelid species have antibodies with an interesting and very useful structure, having a single heavy chain variable domain, which we may isolate as a functional antibody fragment, which is even smaller than that of the previously mentioned SCFVs. These nanobodies are useful because, despite having only one domain, they maintain a similar target affinity compared to normal antibodies with a highly stable structure. They may be correctly expressed in bacterial systems, and due to their smaller size, they may be able to bind hidden or cryptic epitopes that other antibody formats are too large to access. Their smaller size also means that they have a greater penetration into tumor tissues and may even reach intracellular targets following internalization into the cells. Thus, nanobodies have a range of potential therapeutic applications. For their discovery, we can propose our in-house VHH Phage Display library, constructed from 57 healthy camelid donors of 3 different species, alpaca, camel, and llama, with a diversity of 1.51 times 10 to the 10 power variants. However, indeed, you might be saying, one potential problem for therapeutic applications of these is the fact that the VHH is not human, and so an anti-drug antibody response cannot be ruled out. Yet, there is indeed a solution to this. As shown here, the structure of a VHH is comprised of a framework with 3 complementarity determining regions, or CDOs, which are responsible for antigen binding, with the rest of the nanobody, the framework regions, playing a mainly structural role. Thus, once we have identified the nanobody with the specificity we require, we can take these CDO sequences and graft them onto the framework of the relatively similar human variable heavy region, with some design and changes, called back mutations, in order to ensure that binding specificity is maintained. We can thus end up with a humanized nanobody, with all of the advantages that the format brings, without the disadvantage of a non-human antibody. And while we tend to think mainly about human therapeutics, many people across the world have very dear pets who are afflicted by diseases such as cancer in the same manner as us, and to which the same therapeutic principles may apply. Thus, to cater for these veterinary applications, we have developed our own dog and cat Phage Display libraries, again constructed from a large number of healthy donors from a range of different breeds, in order to reach a high diversity of over 1 times 10 to the 10 different clones. Now, I will move on to something adjacent but equally important topics related to Phage Display discovery projects. As getting the sequence, of course, is not the end of a Phage Display project. To make the best possible therapeutic antibody, a lot of work may need to be done expressing, testing, and engineering the sequences obtained in Phage Display, for which we also offer a range of relevant services. Since in Phage Display we work with antibody fragments fused to viruses, the end product of a panning is a sequence rather than an antibody. Thus, the follow-on recombinant antibody expression is very important. For this, we take advantage of our highly developed recombinant protein production capacities here at Proteogenics, where we can take the antibody sequence that you have obtained in Phage Display, carry out gene synthesis, transfection, and then transient expression in the expression system of your choice. The system of choice to express recombinant therapeutic antibodies is, of course, normally a mammalian system, for which we can propose our extensio expression system, which is a Chinese hamster ovary cell line, which we have engineered ourselves for outstanding high-yield recombinant antibody expression. As a lot of different clones may need to be tested following expression in full-length recombinant format, we can propose our high-throughput antibody production process using the extensio system, where we can express hundreds of candidate sequences in parallel, reaching quantities of up to 2 mg per monoclonal antibody tested, allowing for efficient testing and selection of a large amount of candidate sequences. Then, of course, once a suitable candidate with appropriate activity is identified, sometimes a higher affinity version of the antibody may be required. For this, we offer our Phage Display-based affinity maturation process, whereby we intelligently design a mutated Phage Display library with mutations designed to improve affinity, with these mutants being synthesized en masse in order to construct a synthetic Phage Display library, which is then screened for higher affinity binders, which are then sequenced and tested in detail using techniques such as SPR, ELISA, and seq-HPLC. While earlier I discussed anti-cancer therapeutic antibodies that can have a direct effect on inducing tumor mortality by blocking a ligand receptor interaction, not all tumor markers may be exploited in this manner. Often we identify a target cell surface receptor on a type of cancer to which binding will have no such direct effect to induce tumor cell death. Thus, while the antibody is still necessary for specificity to the target cells, the killing effect must be obtained elsewhere. For this, we can offer the development of antibody drug conjugates, whereby a cytotoxic chemotherapeutic agent is covalently joined to the antibody via a chosen linker. The toxic drug may then be internalized and released inside the cell, providing a specific cytotoxic effect against the cancer cell type of interest, with minimal damage to the healthy, non-cancerous cells in the organism. Thus, for this, we offer our antibody drug conjugate development service, where, with the client, we may select the most suitable payload and linker for their application, carry out the required conjugation in a test pilot quantity, with assessment of parameters such as the drug-to-antibody ratio, followed by delivery of the ADC to our clients for testing prior to larger-scale bioproduction and conjugation. For other applications, one may wish to develop what is known as a bispecific antibody, which are artificially engineered antibody constructs with different antigenic recognition for both of the antibody arms. This may be used, for example, in cancer therapy in order to direct an immune cell to a cancer cell, which will then be encouraged to kill the tumor cell. There is a plethora of different bispecific antibody formats to choose from, with bispecific fragments such as BITE, DIBody, and DART, full-length bispecific IgGs such as CrossMab, kappa lambda bodies and duobodies, and fusion protein constructs such as Fab-SCFV or the trivalent Dock and Lock. It can be a bit overwhelming and confusing when confronted with this amount of choice. Thus, since choosing from these different constructs and designing them is no small task, Here at Proteogenics, we offer our great experience in this domain for your bispecific development projects, where we will study your project, help you pick the most appropriate design for your application, then design your construct in the quantities you then design and produce the construct in the quantities you require, along with our potency assessment of the produced constructs in order to verify that the desired binding affinities have been maintained in the new format. So I hope that that this has given you a good overview of the range of services which we have to offer to the domain of therapeutic antibody research here at Proteogenics. I, of course, couldn't cover everything, but we have a fully developed capacity for every step of the pipeline in therapeutic antibody development, from gene synthesis and antibody production to lead discovery via hybridoma, phage display, or single B-cell screening, right through to lead optimization and engineering, as well as the production of your necessary quantities for antibody for preclinical and even clinical studies, through the development of large-scale, stable producing cell lines. Thank you very much for listening to my talk. I hope I've left you with some useful knowledge, and I am now, of course, happy to take any questions that you may have.
Fantastic. Well, thank you so much, Conor, for a great presentation. With that, we'll move right into the Q&A session. And as a reminder, if you do have a question that you'd like to ask, you can submit it using the Q&A tab on the right-hand side of your screen. All right, Conor, we'll jump in with a great question here. So what are the benefits of using phage display to generate monoclonal antibodies compared to some other techniques?
So indeed, in addition to the advantages that I talked about in the presentation about going directly to fully human antibodies and thus not having to humanize a mouse antibody, there are, of course, other advantages such as the ethical advantage of not having to use animals, the speed, as a phage display panning can be done rather quickly in 3 to 4 weeks. And of course, there is no limit, essentially no limit to the targets that we can use. So we can also pan against a highly toxic candidate that would be impossible to inject into an animal, or unethical to inject into an animal.
Fantastic. A few questions here about the libraries. So are the cancer libraries available for purchase? And we can combine that with another question here. What are the different libraries available for purchase through Proteogenics?
Okay, indeed, we do offer our proprietary constructed phage display libraries for sale. However, they have a very large and almost incalculable value. So they are rather expensive, and they do come with certain restrictions on the intellectual property rights of any sequences to be used for commercial purposes, etc. But indeed, theoretically, they are up for purchase.
Excellent. And then also another question here. What are the different libraries available for purchase at Proteogenics?
Indeed, sorry. Yeah, you did say that. So we have the ones that I mentioned in the presentation. We have our cancer and autoimmune libraries, our naïve human library. We have the VHH library, which I also mentioned, and we also have a rabbit-based phage display library, and we can then, of course, construct any libraries from mouse or rat or any other species that we have access to, or from material that a client gives to us.
Excellent. All right. Thanks a lot. Another good one here. So do we have to send you the antigen for panning in the project?
You can, yes. We do a lot of both. We have a lot of clients that have a very specific antigen that they possess already, but we also have a very well-developed recombinant protein production capacity. So we do a lot of the antigen production for our projects themselves.
Fantastic. And how do you properly coat peptides or small molecules for panning?
So for the coating, indeed, of small molecules, it coating them themselves is not very efficient due to the lack of adsorptive interactions. So for this type of project, we always conjugate these to some protein carriers, which will allow them to be effectively coated. And in those cases, to avoid enrichment of binders against those carrier proteins, we will do several different ones, which we will then change over the rounds to avoid enriching against them.
Fantastic. A few questions about antigen here. So how much antigen would I need to provide?
About 1 milligram is more than sufficient for a Phage display campaign.
Excellent. And what types of antigens can be provided as well?
It's almost anything, I would say. Anything that can be any peptide sequence, any full-length protein. And for small molecule antigens, I would say the limitation is chemical with regard to whether we can conjugate it to a carrier or not. But if you have a good team of chemists behind you, they can probably do some magic on that and get it conjugated.
Perfect. Really good question here. Is it possible to select antibodies against one specific protein, but not another protein?
Yes, absolutely. So for that, we use a similar strategy to what I explained in the presentation with the ligand receptor complex. So there we deplete against that complex. But say if you have a project where you have two very similar proteins and you want your antibody to bind one and not the other, then we can take that negative protein and do depletions of the library so that we're only enriching for antibodies that are positive against the unique part of your protein that will render the antibodies you get out target specific.
Right, makes sense. Do you provide guarantees on your Phage display projects?
Yes. So for most of our projects, of course, it is target dependent. We do have a financial guarantee that we will get at least one binder out. Yep. And if we don't, then you only, of course, pay a portion of the cost of the project to cover our costs.
Excellent. Yeah, it's great to hear. What's the speed, or how fast can you perform a Phage display project?
It's about 3 to 4 weeks. Of course, it varies, especially if there's a lot of different screens that need to be done or complicated cross bannings. But 3 to 4 weeks, we can generally get a sequence. Yeah.
Excellent. Another good question here. So, in your opinion, what bacterial strain is best suited for this antibody Phage display?
So for these, we use TG1 bacteria. I believe the reason that they are commonly used in Phage display is because of the longer pili, which allows them to be better infected by the phages.
Excellent. Question here about storage. So how long can the phages be kept in -80, and how many times can they be thawed and frozen before they lose their infectivity?
At -80, they can be stayed. If we store them at -80, we have to store them as a glycerol stock. And this can go from 6 months to up to a year. The freeze thaws are very, very severe on phages. I think we can lose 10 to the 10. We can lose a power diversity with one freeze thaw. So this is absolutely something to be avoided. And it is probably better to stock phages infected in the bacteria and freeze that.
Right. Yeah. No, that makes sense. All right. How do you transfer the IP of the identified sequence binders?
Yes. All the IP from the sequences that we identify in our pannings is 100% the property of our clients.
Excellent. And how many Phage display pannings can be performed in parallel?
We have the capacity for several dozen, I would say. Hard to put an exact number, but definitely several dozen at once we can do. Yeah.
Awesome. Great to hear. All right. So this person writes, I'm assuming that many customers may not want to share their IP with the CRO. So how would you go about this?
We are very used to signing non-disclosure agreements if it is absolutely necessary for us to know the identity of the targets. However, it is theoretically possible for us to do a panning blind if it is absolutely necessary for the client. This is opposed to say hybridoma, where we're injecting an animal and it is 100% necessary for us to know whether what we are injecting is toxic or not for animal welfare reasons. But we're well used to handling projects with a high degree of confidentiality, and we've never had a problem.
Right. Yeah, it makes sense. So how long does it take to produce the monoclonal antibodies?
So if we're talking about from the end of the project where we get our sequence and the recombinant production, that takes about 4 to 6 weeks in our extend-show expression system, which will get a relatively high yield of in a small format.
Excellent. So we talked a bit about other, the libraries that you already have available, but do you also offer custom library construction?
Yes, absolutely. So I think the species that we have available for custom library construction are mouse, rat, camelid, and rabbit. So we can do that either as naive, where we can just take one of the animals and use PABMCs to construct the library, or what is more common, I would say, is to construct an immune library against an antigen that a client gives to us. Then we'll immunize, and you'll get a nice pre-enriched Phage Display library against your targets.
Excellent. All right, so maybe a future-facing question, Connor. Are there any specific developments or sort of research avenues that you're particularly interested and excited about moving forward?
Absolutely, yes. We are definitely looking into as it's a bit chic at the moment, artificial intelligence, in order to be able to better identify what are the best sequences, and in the end to maybe even avoid using a Phage display itself. We're just having our own algorithm that can generate an antibody sequence that we will then be able to express and test with a high degree of certainty of binding. That is maybe not ready for the end of the year, but we're definitely working on it, and we're quite hopeful about what we will get out.
Excellent. I think we'll end on that high note there. But Connor, thanks so much for the great presentation and all your insights in the Q&A. It's really been a pleasure talking to you today.
Perfect. Thank you very much, Liam. And thank you to everybody that listened and stayed for the Q&A.
Yeah, definitely. Big thanks to the audience for participating here. And in closing, we hope you enjoyed this Scientist.com and Proteogenics webinar, and we'll see you again next time. Have a great day, everyone.